extremophiles in picture to look into the enzymes which are stable. And why enzymes from extremophile? Because as I already told you, they are stable even at extreme conditions, may it be temperature, pH, or high salt, or any other extreme condition. So life in extreme environment, again, these extremophiles are divided into different groups based on what kind of ecosystem they are residing. So for example, I told you hot spring. So thermophiles are the organisms which are surviving in hot conditions, and then acidophiles are in acidic environment. Psychrophiles, like I visited Antarctica recently, and all the organisms which are living in those ice sheets or in the areas where there are very low temperature, all known as psychrophiles. So then again, there are halophiles. Halophiles are also known as the group of organisms which love salt environment, like sea salt. And there are other saline ecosystem like Sambar Lake also. I'll show you some of the data from our study on Sambar Lake. So in case of an organism which is showing more than two different kind, which is surviving in two different kind of conditions, like for example, an organism which is psychrophile, which can survive in cold condition but also can survive in halophilic condition, are known as polyextremophiles. Means they have the properties to withstand the two different extreme environments. And this also demonstrates that life flourishes even in the harshest of location, because if you see the type of conditions they are surviving, it's more than what we human beings are living. I mean, these are very harsh conditions, and it also provides a clue that life arose in hot environments. Thus, this particular ecosystem can present as a model of ancient life on Earth. So if you look at the contribution of extremophiles, it's a huge contribution. They have a contribution in biofuel. They have contribution in production of lipase and these amylase enzymes, which are stable in industrial processes. But I'll just give you one such example of DNA of extremophiles where they have contributed a lot. If I'm sure you have heard about Thermus aquaticus, and I'm sure you have also heard about PCR. So when you set up a PCR reaction to amplify the DNA or a desired gene, you need to add the enzyme which is required for the amplification of a particular product. So this product has come from an extremophile called Thermus aquaticus, later Pyrococcus and Thermococcus as well. And imagine if these organisms were not discovered and the processes from these organisms were not discovered, we would have never set a PCR, which is automated PCR. And you can see that this patent has earned its right holder over $2 billion as a royalty. So imagine the potential of these extremophilic microorganisms in the industrial level as well. But here, what I want to point out is it is very, very difficult if you look at the organisms which are surviving in environment to cultivate in laboratory conditions, because the one thing is we cannot provide them the desired condition. Probably there are micronutrients which are present in the environment. We are unable to detect them, and we are unable to provide them in the media, even though we customize the media. So it has been noted that not more than 1% of the microbial communities which are present in an ecosystem have been cultivated so far. So it is very, very important to know where this 99% is hiding. I mean, why we are unable to cultivate these organisms. So to give the answer of this particular question, I'll come to you later. But before that, I want to tell you that whatever we isolate, whatever microorganism we isolate, we go through several processes of identification of microorganism, because it is very, very important to identify microorganism, to know about the phylogeny or the lineage of these microorganisms with other organisms which are present there. So it's a very basic characterization which we used to do earlier. to look into the type of microorganism these are, and there are certain journals, taxonomic journals, which still require these kind of data for the characterization of a new or newly discovered microorganisms. So morphological and microscopic features, then biochemical features like catalase activity, utilization of sugar, etc. And then comes the molecular feature, which is more stable way of looking into the and more accurate way, I will rather say, of identifying a particular um, microorganisms. But fine, we want to do the molecular feature of a particular microorganism, but how? What we have to do in case of identifying bacteria or archaea to get an insight into the identification of that particular organism. So there is this gene which is called 16S-R RNA gene, which is present as a different conserved region and variable and hypervariable regions. It has a length of approximately 1500 base pair. And why we prefer, because all the microorganisms does have this particular gene, and the most important thing is it is a very stable housekeeping gene, and also known as a molecular chronometer for the identification of microorganisms. So based on 16S rRNA gene sequencing, we further go for the identification of particular bacteria or archaea. Although in some of the cases, if you look at the genus Bacillus, we still have to use some of the other genes, housekeeping genes, to get a clear or get a better resolution of the identification up to species level. But 16S is the best marker so far for the identification of microorganism, because it has a long stretch and it gives a good information about the taxonomic lineage. And so why do we need to even if we do cultivation, where we lag behind? I mean, like I told you, that 99% of a microorganism which we culture, which we process in the plate, are uncultivable, because only 1% can be cultured, or maybe less than that. So how to get an insight of these kind of microorganisms? Because it is very, very important to have these microorganisms known, so that we can draw a conclusion. Because just based on one or two or even 10 microorganisms, we cannot conclude that what kind of role they are playing in that particular ecosystem, until and unless we don't know the whole community structure over there. So it's all started with the high throughput sequencing technology, which has helped the microbiologists to look into the complete uh, microbial communities which are residing in a particular ecosystem. For example, if you're playing in a garden, you want to know what kind of microbes are there. If you want to culture those organisms, you probably land up culturing 10 or 15 microorganisms, or maybe if you use 50 or 60 media, you may land up in a situation where you have 100, 200 isolate, but still they are going to be, majority of them are going to be similar kind of microorganism. But the majority, 99%, we won't be able to know about that. So then it started with the first generation sequencing, second generation, third generation sequencing. I'm not going into detail, but just I want to tell you, like, the way it is written, that first generation, second, I mean, it has more throughput. So it is giving more and more data. In the second generation sequencing is more accurate and gives more data compared to first generation, and likewise third generation gives you more data compared to second and first generation, and has more accurate and longer reads. Before I go into more specific area, I just want to make sure here that this is probably something which, for MSc students, this may not be uh, very understandable at this moment, but I suggest you to go through this particular sequencing technologies. There are many papers which are available. Go through these papers and read the, how it started, the evolution of sequencing. There is one paper in genomics, you can probably read that, and this will give you a very nice information how the sequencing has evolved from a very short stretch and longer duration to short duration and longer reads and more data. I'll probably give you more glimpse on that if I have that slide. Probably, I think I have that slide. Yes. So yeah. So generally, what is the strategy? I'm just telling you the overall approach of looking into the whole community structure. So what we do is we collect samples and then we do community DNA extraction. And earlier we used to do cloning. I'll also give you a few information about cloning. And then we also used to do DGG and why these are absolute techniques now. Why we don't use these techniques? Why we use direct sequencing techniques these days? So I'll give you those important information. So this is the overall graph which represent how a microbial ecological studies can be performed. And here I would like to say you that earlier, very I think it's more than 1.5 decade back, we used to do denatured gradient gel electrophoresis, where we used to put the sample in the gel, acrylamide gel, and based on their size and based on their GC concentration, they move on the gel, and based on that, we pick up those gels, sequence them. But the drawback of this technology is that we can pick up 10 to 15 band, and there are certain bands which are overlapped. For example, if you have a 16S RNA gene which you are running on a gel, has almost equal GC content compared to a different species. So the possibility of these particular these kind of bands is that they may overlap, and you will get a false data out of that, because you are not able to distinguish those bands. So, and again, here you may not be able to go beyond 10 or 15 bands, and again 10-15 band is not something which will represent the whole community. And then it came the clone library construction, where this is also no one is using these days for microbial ecology study, because it has drawback, because when you clone a particular gene in a clone and then you grow it on the plate, probably you have 100, 200, 1000, 2000 clones which are present on the plate. You may able to sequence those genes, you may able to get some of the information of the microbial communities which are present there, but there is possibility that you may not be able to pick up those clones which are very rare in the community, and ultimately leading to a very false impression of what kind of community is present. So the observation may lead to a different uh, um, results, where you may not get very accurate results. So that's why this high throughput sequencing came. So what in this case, what you do is you do the whole DNA extraction. So each and every DNA which is present in your sample, you have to generate libraries, and then you straightforward do the sequencing, and based on the sequencing you just go and do the bioinformatic analysis, and you will get to know the kind of microorganisms which are present there. So for using any DNA sequencing technology, it's very interesting to know that the technology should be fast, it should be accurate, it should be easy to operate, and of course it should be cost effective, because if you look at the first human genome sequence which was performed, it I think utilized millions of dollars to just do one genome sequencing. And if you look at the present technologies we have, I think we can do that genome sequencing even in $10,000. So imagine the differences in cost effectiveness and accuracy of a particular technology. So DNA sequencing is not just important to look into the microbial diversity or microbial communities which are present there, but they also give us an information about the functionality of these particular microbes present over there. Their structure, of course, they will give an evolution, but they also give us other information like genetic disorders and other diagnostics doing the DNA sequencing. So evolution of sequencing, I was telling you, it over the last few decades there was a remarkable improvement in the sequencing. So if you imagine doing sequencing in 1980, if you have to do sequencing, you will get 1 KB of data per year in 1980. And now in 2021, imagine that you are getting 100 GB data per day. So this is how the revolution has started for the sequencing, and this is where we are, and this is how things are working in microbial ecology or microbial, or for that matter, for any sequencing based studies. And also, like I told you, in case of the Human Genome Project, there were millions of dollars which were spent. But if you look at today, if you want to go for a genome sequencing, probably you need $1,000 and you will get a genome sequence done. So this is how the technologies has come up, and this is how the cost effectiveness has improved our way of doing analysis, and also gave an opportunity to the laboratories across the globe who are not having much funding but still can do these kind of studies. And yes, absolutely. So now we have all the sequencing data with us. Everything is with us. But what to do now? Because we cannot just rely on the sequencing data, raw files, putting in the folder. So then we have to perform the bioinformatic analysis. I'll not go into details, but I'll just give you a glimpse that once we have the raw data, we process this particular raw data for quality control, and then OTU based analysis we perform. So OTU based analysis is basically operational taxonomic units. So one particular sequence which is associated to particular sequence in the database is basically one organism. So this is how we perform, and we also can perform community characterization, functional metagenome also we can perform. I'll show you some of the data which we have done using predictional functional metagenome using predictions, and we can look into the diversity, phylogeny, and taxonomic composition using bioinformatic analysis. And it takes a lot of time to perform bioinformatic analysis for this much data, because we have data in GBs, so it takes a lot of time to run these commands and get the data in the way we want. So we have performed one study in Soldar Hot Spring during my PhD, and we have collected samples. I just want to give you an overview of why high throughput sequencing gives you more accurate data and more information about a particular ecosystem. So here you see we have published one paper in 2009 where we can only culture 17 isolate using multiple media, and these isolates were belonging to 2 genera. And then we again performed, like I told you, DGG and clone library, which are also, they don't give you much of the information. So in this case, we could only be able to detect 6 genera using this clone library and DGG analysis. However, we did a deep sequencing analysis, the high throughput sequencing, which I'm telling you, where we found 33 OTUs. Means these particular sample which we processed for DGG and clone library, we could be able to analyze, observe that there are 33 different kind of genus which are present there in this particular sample. So imagine the difference in the technology and how they are giving you the accurate data, because if we would have never performed this, we would have never got an opportunity or got an information that this particular sample does contain 33 different kind of OTUs compared to what we have performed here. So this is the beauty of high throughput sequencing. It gives you a very robust, very accurate, and very depth data for your analysis. So again, we performed one study. I kept this slide especially for the sampling purpose, because most often what we do is we do sampling and we collect sample, bring it to the laboratory condition. And these 2-3 slides are especially for those people who are doing a lot of cultivation studies, and what could be the reason of delay, and delay in sample processing may lead to the cell viability issues. So it is very, very important to devise a strategy that how you're going to perform sampling and how you are going to do your cultivation studies. So here I just want to point out that we have collected samples from Sambar Lake, which is a salt lake. It has a very high salt concentration over there. And what we did is we used two different approaches over here. One, we did the on-site cultivation in three different media based on our experience of cultivating the halophiles. So we did on-site cultivation, when then we did cultivation in the laboratory condition after collecting sample, bringing it to lab, and then again processing these particular samples in the laboratory condition. So here, if you see, there are seven, I guess, one, two, three, four, five, six, seven different kind of genus which we could cultivate for only from the on-site cultivation. So here again, you can imagine if you're sampling or if your approach is not very accurate, you may lose these seven different type of genus which we could able to cultivate in case of our on-site cultivation approach. So it is very, very important when you're going out for sampling. It is very, very important to note down what is the objective of your study. And if you want to do cultivation study, you have to perform multiple approaches so that you can cultivate majority of the microorganisms in laboratory conditions. So in this particular study, we were able to, I think around 13 of these microorganisms were novel, which we could isolate from the on-site cultivation approach. And here you can see three of them were bacteria and rest were archaea, which we could isolate from the on-site cultivation approach. And I think two of these organisms we have already published as a novel species in the journals, which are mainly accepting these kind of work like taxonomy and discovery of novel microorganisms. So this is why I kept this particular slide to devise the strategies for sampling, which plays a very, very important role. And then we have also did some study on the Arctic fjord I think the samples were collected in 2018 or somewhere like that. And then what we did is these samples were collected from different depths. I mean to say the surface of the sea level, but there are different depths over there, the stations which are present there in the Arctic. So here we found that there is definitely a differences in the kind of microbial communities which are present at different depths. And also, if you look at the kind of archaeal community specifically, is also very different. If you see in class 9, there is a different kind of microbial communities, and likewise in case of 5 and 1. So the idea here is to show you that this is how the high throughput sequencing can give you the result even in case of a different kind of samples you have collected. And it also gives you an imputational or functional potential of these microorganisms by looking into the gene abundance. So in this case, what we did is we also look into the gene abundance, which are using 16S RNA gene database. We found that these organisms do play a lot of role in ammonia and sulfur cycling, which is very, very important because ammonia and sulfur both are toxic for the environment. So this is how this particular type of microorganisms, especially archaea and bacteria, are performing a lot of biogeochemical cycling, and these are helping in utilizing ammonia and sulfur for further ecological balancing. And this study was published recently, I think last year, in the journal Genomics as a short communication. And then recently we have one study which we conducted with IITM Pune during our expedition in Antarctica. So we performed some of the sampling over there. So for the 20 days, we performed sampling every alternate day. Although this is a very small time period to assess a particular changes in the microbial communities based on the abiotic factors, but we were more interested, since this was a very short trip and we were unable to perform the yearly based pattern of changes in microbial communities. But here we were more interested to look into the impact of slight change in temperature, because there is slight change, not even slight change, but there is climate changes which are happening in Antarctica as well. The temperature is even going to 7 degree to a 9 degree these days. So we were more interested that even if there is a slight change in temperature, is there any impact on microbial communities? Because the natural resilient microbes which are present there in the Antarctica needs to be conserved before the temperature goes high and we lose all the microorganism which are present in those ecosystems. So here what we found is, when there was a rise in temperature here, if you can see, there's a complete different pattern of microorganism which we can see over here. And there's a slight very slight rise in the temperature from zero point 0.5 to 3. And here you can see there is a complete different microbial community structure over here. And if you can see, there are firmicutes which are getting highly abundant compared to the other microorganism. So this was the only difference which we find. And even if in this case you can see in the sample 7, there are different community structure which are changing because of the temperature. Although the UVB radiation was quite stagnant, there were no changes, but the impact of temperature has a direct impact on the community composition. So this itself, the study basically shows that how important it is to conserve the microbial diversity of any particular ecosystem going through a climate change issue. So this all is treasure. All these microorganisms which are present there in any ecosystem is a treasure. We have to cultivate these. We have to bring them into the laboratory condition for future use. So this is the take-home message from this study. And also we did some functional imputation metagenomic over here. I will not go into detail of this because you may not be able to get the clear picture of what exactly is going here in this particular slide. But just to give you a brief introduction, so because of the harsh condition, these microorganism has a direct impact of UV on their cells, and there could be a possibility that there is definitely going on. There is a possibility that there may be a damage in the DNA, which is very, very important for the functionality of a particular cell. So we looked into different metabolism. They also do methane, nitrogen oxidative, and sulfur metabolism. That is basically a utilization of a substrate. But here they also have a gene which are very highly abundant related to the base excision repair, mismatch repair, which all these genes are responsible for the DNA repair mechanism in these kind of microorganisms. So when I was talking about cultivation of particular microorganism and bringing them into laboratory condition and conservation of these particular microorganisms, then I will also take you to our repository, which is NCMR, National Center for Microbial Resource. We have more than 200 250 or you can say 2 lakh cultures which are present with us at the moment, and we are one of the world's largest microbial repository here. So I request you all, whosoever is generating any kind of microorganism of any use, or whether it's a very precious microorganism, if you don't feel it's a precious, please do submit your microorganism to our repository so that these can be used freely for can be available for other researchers, where they can utilize these resources and look into the various aspects. Because if I am working on a particular aspect, um, I may not do everything for a particular bacteria. So that's why it's very, very important to deposit these microorganisms in these repositories so that they are preserved for a long-term duration and they are also available. For example, I'll give you an example of Streptococcus strain, which was isolated from a child in 1958 or something. I forgot the year. So that strain is still there in the repositories, and that strain is actually a model for looking into the research studies related to urinary infections. So you can imagine your one organism may deposition of one organism may change the whole scenario, and maybe of very high importance, which you may miss otherwise by discarding or throwing it. Um, so I request you all to deposit your organisms or any biological material to our repository. And then we also have a different kind of culture deposit service. We do send culture to different research organization or universities who are interested to do some sort of research on these isolates. So if you want to look into these microorganisms, what kind of organisms we have, you can go to catalog and look into the kind of microorganism, and look into the kind of ecosystems we have procured these organisms. And we also do genomics and metagenomics kind of analysis for the people who are interested in doing such kind of analysis. So with this, I think, take my presentation towards the end, and I would like to thank our director NCCS, who has always there were actually 3 directors when I joined here. The present director is the 3rd director. So I would like to thank all the directors, because they have helped out of way to uh, let my research go with full flow. Dr. Yogesh Yoche is always there to help me. Kunal Jani was my student who has presently joined a research group in Czech Republic, and he has also done a lot of work with me. And Dr. Swapnil is currently working with me. He has just defended his PhD and looking for some postdocs. And I have a lot of master students who have worked with me, and they have also contributed to various articles which we have published. So it's always nice to see master students contributing in the form of publications. And I would like to thank India Alliance for funding DBT, NCCS, of course, for funding German Science Foundation, DBT, TAWAS, INSA, and in ICMR for funding my PhD, and also MOES for funding my Antarctic trip. Thank you very much.
Thank you, Dr. Avinash Sharma. Are there any questions?
Yeah, I'm Dr. Nandakumar Kamath. I would like to ask Avinash. Dr. Avinash, can you hear me?
Yes, sir. Go ahead, sir.
Yes, please ask.
You see that you mentioned about the impact of delay in on-site and off-site culturability of these extremophilic organisms which you tried to isolate and were successful from Sambar Lake, which is a hypersaline lake, I believe. I mean, what I couldn't get was the explanation, means what is the scientific explanation? It has something to do with the ecologically specific parameters associated with this, or is it something like, viability of the cells which are so sensitive that the little delay out of the in situ environment would cause problems? Means we need a scientific, the why is more important for me than how and what was done. Why is it so that
Great.
on site and off site, you see there is a difference? What is the reason in terms of the cellular characteristics of these extremophiles?
Okay, so I'll answer your why. Um, so the idea of doing this particular exercise is as we have performed a lot of studies, and we were more interested to look whether supplementing a particular sample with a particular media, is there a chance where we can capture more microbial diversity? Because, and it does, I mean, practically, if you look into this a particular hypothesis, it is very important to give some substrate to the organism before you collect these samples, bring it to the laboratory. There's a delay of a lot of hours you are spending in transporting these samples. So we were more interested to look into the delay in sample processing, basically, it has an impact. So that's why we have done a comparative analysis, not the analysis, but a comparative approach to see whether we can capture, because there are many organisms which are very sensitive to temperature, and they need they don't have the regulation or the cellular regulation which can keep them inactive but again active form when given the media. But there are organisms which are easily getting dead because of long or delayed sample processing. So that was the idea of doing this particular exercise.
Thank you, Dr. Avinash. Any more questions? Yes, Dr. Jyoti. Dr. Jyoti. Okay, Dr. Flory.
Yeah, hi. I have a question. Sir, you spoke about metagenomics, and I believe that you all are the experts in metagenomics. My question is, normally, sometimes you are able to isolate some organisms which are non-cultivable. Okay, how much is the success of using these particular organisms for a particular project? For example, for some bioremediation or something like that. Is there any success, or is any success being reported so far?
Sorry, I was on mute. So you mean to say the culture you have in hand, or you want to talk about the metagenomics?
Suppose you isolate some particular organism by metagenomics, okay, I mean genes of the whatever, and how successful would you be in using this particular genes for bioremediation?
No, using metagenomic, we cannot culture a particular organism, because it's just an information, gene-based information you are getting out of this particular sample.
True, true.
Yeah.
How that my question is exactly that. So how useful
Yes.
is this particular
So the data is useful in a different way. I'll put in it in a different way. So here, what will happen is, see, when you are collecting some sample. Let us keep two scenarios over here. One scenario is, you have a sample with you, and you want to do it the isolation of particular organism from this sample. But on the other hand, you have the metagenomic data with you, and now you can focus on that particular sample, that okay, these are the kind of microbial communities which are present there, and this is the specific media which is used to cultivate this particular organism. So this is how this sequencing data is helpful in, devising a strategy to generate or to develop a media where you can put specific substrate which is suitable for a particular organism. So this is how this sequencing data is helpful in cultivating the yet to culture microorganism. So this recent study which I am doing in our Wellcome Trust is basically on the high throughput cultivation of these microorganisms. So what we are doing is, we are first looking into the total community structure, and then based on that, we are giving different substrate. Like in case of a particular specific group which can grow only on LB, we'll give LB, keep LB as a media for the cultivation of those microorganisms. So likewise, I mean, we are also customizing different kind of media for the isolation of these microorganisms. I hope this is what you were asking, right?
somewhat. Yes, okay, thank you.
Yes, Dr. Avinash, there is another question by Dr. Jyoti. Can I read it? What is the efficacy of metagenomic analysis?
Efficacy of metagenomic analysis is, I will surely say that it's not 100%, but it has high efficacy compared to the other technologies, specifically looking at the identification of what particular microorganism from an ecosystem. But definitely this doesn't give you 100% coverage. There
Okay,
is nothing perfect.
yes, okay, thank you. Any other questions from the students? from the research scholars? Okay.
I think there
So
is one question. Okay, it's done. I think.
it's done.
Yeah.
I read it out.
Yeah.
Okay.
There was one question earlier. I don't know. I cannot see in the chat box, but there was a question. I think maybe I have missed it. Okay.
Okay. So if there are no more questions,
No, I have one more question. My mic was on mute. Hello. One more
okay,
fundamental
sir.
Okay.
question.
Okay.
Yes, sir, you can go ahead.
Professor, I have, because you have a lot of experience, I have seen some of your papers, especially nasal bio, this one microbiome of SARS infected patients. You also worked on yeast, I believe, right? Hello.
Yeast, I have not worked much, but we have done some
Yeah,
study
some alcohol
collaboration.
beverages and all. I saw
Yeah,
some papers.
yeah.
So it's a very interesting and extremely diverse study that you have done from your presentation,
Thank
and
you.
it's good. My
Thank you.
fundamental question is, I'm a microbiologist and I'm guiding a student. We use a Winogradsky technique for enrichment of samples, Winogradsky technique. Hello.
Okay.
So right now the issue is like this. We identified a very efficient sulfide oxidizer,
Okay.
and sequencing shows that, it is not matching with what is deposited in gene bank. Now the question is, how to prove that it is a novel taxa? Means what are the requirements? Because we said that from Sambar Lake and other places,
Mm-hmm.
you have raised some novel taxa. So what are the
Yeah.
requirements? Because our similarity and everything doesn't match with anything in gene bank. But
So you have done 16S for that?
yeah, correct, 16S, and it is not matching with the 95%. Even 95% is not matching with. It is showing all unrelated things which have nothing to do with sulfur oxidation. Understood. So it's impossible from
And
other.
and if I ask you from where you have done this sequencing?
Yeah, we have done it from an outsourced, reputed, that laboratory.
Okay.
I can't disclose you that, but
No, I don't
generally,
want it the name of the company, but I just wanted to know whether it was a research organization or a private company.
yeah, it was a private,
Okay.
reputed research organization from where earlier also we have validated the data and published it also.
I can only help you if you can send me, if you I mean, if you're fine with that, you can have my
No,
email.
we are not interested in sending. We are collaborating also with I am just asking you, what are the requirements? Is it
No,
GC
this
content? Is
19
it
this
DNA hybridization? Is it
no
fatty acid analysis? What is it?
no this see, the first thing is 16S RNA gene sequencing, but if it is showing 95%, one thing is your bacteria is novel. Okay.
Yeah, it is less than that. It's
Yeah,
not even
even if
95%.
it is less than that, it's a novel organism. The other thing why it is showing less than 95% is probably the bad sequencing. So that's
Hmm.
what I was telling you, that if you are interested, if you have experts, you can share the AB1 files with them and they can have a look into those AB1 files. If they have this quality issues, then you have to redo the sequencing. If not, then you can go ahead and look into the and different kind of chemotaxonomic factors like fame analysis. You can also go ahead for minaquinone and also do
We are
polar
thinking of whole genome sequencing.
whole genome you have to do, because if you want to perform you want to describe it as a novel taxa,
Yeah,
you
correct.
have to have genome sequencing. It is mandatory from 2019 onward, so you
Yeah,
have to
but
go for.
problem is only into 10 coverage they are asking, and 100 times coverage is not possible. Into 100
But
full coverage is not.
draft genome is fine for
Okay,
taxonomy
draft
analysis.
genome we can do.
Yeah.
I will contact you separately, sir. Right now,
No
I don't
problem.
want to waste your time.
No,
Thank you
that's
very much.
okay. No,
Thank you
thank
very
you,
much.
thank
At
you,
later date, I will contact you.
thank
Hello.
you, thank you.
Thank you, Dr. Avinash Sharma, for addressing the questions. Any more questions from the participants?
It seems there is no question, and I'm sorry I was not quite audible today. I'm not feeling well from last 2 3 days,
Oh, okay.
so I'm sorry about that. If there was some
No, you
voice
were very
issues in
audible.
between. Okay,
Yes.
yeah.
Okay, so I would like to thank Okay, I would like to give vote of thanks on behalf of the team of the national webinar, Technological Advances in Applied Microbiology, organized by the Department of Microbiology, Goa University. I express my humble gratitude to Dr. Avinash Sharma for the informative session on understanding life in extreme environments from conventional cultivation to high throughput sequencing. Thank you, sir, for giving us a brief introduction on the role which microorganisms play in our day to day life and why it is important to study life in extreme environments, highlighting the contributions and potentials of these extremophilic microorganisms in molecular biology, especially PCR. Also, why are we able to cultivate only 1% or less of microorganism, and how we can use high throughput sequencing for studying the remaining 99% of the yet to be cultured microorganism? Belonging to the family of microbiology, we thank you for highlighting the importance of sampling approach and processing of the samples so as to obtain maximum cultivable microbial genera. I am confident that the participants are definitely going to benefit from your scientifically sound talk, especially the way you made it clear that the evolution of sequencing from the basic to the high throughput sequencing. The master student as well as the research scholars would definitely be benefited with respect to the various techniques employed in microbial taxonomy. Thank you for stressing on the importance of microbial deposition and letting us know about the world's largest microbial repository, that is National Center for Microbial Resource, NCMR at NCCS Pune. We are grateful to you, sir, for sparing your invaluable time from your busy schedule and enlightening us with your scientifically sound lecture. Thank you.
Thank you very much, and all the best to all of you. Thank you. Thanks a lot.
Thank you. We will take a break now. Follow the schedule, okay?
Good afternoon, sir.
Yeah.
So we will wait for some time since the session was supposed to start at 3:30.
Okay. So, yeah, my voice is okay and everything is audible now.
Yes, sir.
Okay. Yeah, we will wait. I will
Okay, sir.
turn off the.
Thank you.
Okay.
So, shall we start?
Yeah, Dr. Drupti.
Okay, sir.
Yeah, you can start.
Okay, thank you, sir. It is an honor to introduce Dr. D. Balachandar, Professor, Department of Agriculture Microbiology, Tamil Nadu Agriculture University. Sir holds a PhD in agriculture microbiology and has a tremendous experience of over 20 years in the field of teaching and research. Sir is well known for his contributions in the field of soil microbiology, plant microbe interaction, and biofertilizers. Sir has an expertise in rhizosphere engineering for plant microbe interaction, soil health and microbial diversity, and development of bioinoculants in nutrient management and abiotic stress mitigation. To commemorate few of his achievements, so is a recipient of DBT Overseas Fellowship as young scientist during the year 2006-2007 and associateship training at Florida International University during 2007-2008. He has also been an active participant in a Indo-German collaborative project handled by DBT and Federal Ministry of Education and Research. So has published 2 books, 50 research papers, and 20 book chapters. To his credit, he has successfully completed 4 research projects and is leading 2 ongoing research projects, which are funded by esteemed funding agencies like DBT, ICAR, Biodiversity International, and Ministry of Agriculture, Government of India. On behalf of the organizing committee and Department of Microbiology, I take this opportunity to welcome you as a distinguished speaker to this national webinar on technological advances in applied microbiology. I now request Sir to kindly take forward the session.
Yeah, thank you, Ritu, for your nice introduction.
Thank you, sir.
I hope I'm audible, right? Yeah.
Yes, sir.
Yeah, I will just turn off my camera and share the slides.
Yes, sir.
So I hope you can see my slides, right?
Yes, sir.
Okay, so first of all, I should thank Dr. Sandeep and Trupti and all other staff of microbiology who organized this seminar and invite me to give a lecture on recent trends in biofertilizers, right? So thank you for giving this opportunity to share my experiences with you. So um, my presentation would cover these areas, like a brief introduction about biofertilizer and how the biofertilizer development happening in India, and what are different types of biofertilizer, and what is the new developments that are going on, and what kind of constraints that are right now available, and what next about the biofertilizer technology, and a summarized conclusion, right? So even though the list look a little bit more, I will take only 30 minutes to complete about 25 or 28 slides, right? So what is biofertilizer? It's a preparation that contains live or latent microbial cells, and when you introduce through seed or soil or through root, whatever it may be, it has to give some benefits to the crop. And if it supplies to nutrients, then we call it as biofertilizer, which the terminology comes from fertilizer, right? And then if it is increases the plant growth by root colonization, then in general, or the generalized terminology used is PGPR, plant growth promoting rhizobacteria. And if they produce some antibiotics or antagonistic chemicals, and thereby it protects the plant from insects and pathogens, then we call them as biocontrol agents. And without all these three, if they use some direct or indirectly, helps the soil health and fertility, decomposition, nutrient cycling, and so on, then we call as general inoculant. Right? I hope now you will be very clear about what is biofertilizer, PGPR, biocontrol agent, and inoculants. So let me concentrate more on biofertilizers in this presentation. So, as I already said, the biofertilizers are mainly for plant nutrients, but now they are added with biotic and abiotic stress tolerance and soil health and so on. Right. So, I would recommend if many students are there, then I would recommend to read this Microbes Can Help Feed the World, which was published by ASM, and it would be around 35 to 40 pages keynote, which was published in 2013 and freely available in ASM website. So you will find a new dimension in the microbes and how you can explore more for human health and plant health. Right. So this picture shows the overall functions of inoculants and their benefits to the plants. With reference to commercial biofertilizers, and these are the lists which are successfully produced and distributed to the farmers, and they believe that they can give some benefits to the crop. So the oldest one is the rhizobium, and the newest one is the PPFM. PPFM stands for pink pigmented facultative material. So parallel to rhizobium, Acetobacter was also gaining importance, and rhizobium was mainly developed from Europe to US and then comes to India, whereas Acetobacter mainly established in Russia and then comes to India and so on. Azospirillum, I hope everybody knows, it starts from Brazil and comes to India. So like that, several successful biofertilizers are there, and this table shows the commercial available biofertilizers right now in India. Right. So if you come for the formulations, right? So how, in what way the biofertilizers are right now available? This is the historical development of biofertilizer. So in olden days, it was started with clay balls, right? So they don't have any good carrier. So they made a small ball like with the help of clay, and the biofertilizer strains were inoculated and supplied to the farmers. And then it was transformed into alginate and cyst and spore, and finally uh, it standardized or stabilized with peat, right? So peat is the original carrier which was successfully used for biofertilizer all over the world for nearly 25 years. And lignite is in India, we don't have peat, so we alternatively we use lignite for last 25 to 30 years, but now that carrier-based inoculant had a lot of issues. So alternate to carrier, the liquid formulations have come up, and now the liquid formulations and the microbial consortia are now ruling. And what next is the last two. So we may get micro biofertilizer coated seeds, which are when you sow to the field, and the biofertilizer coated on the surface of the seed can inoculate, automatically colonize the root, and they may give you a benefits, right? And then now another dimension in biofertilizer is the freeze dried powders. So it is like a capsule. So you can condense all the cells into a very low volume. So maybe 5 gram or 10 gram per hectare of seed is more than sufficient, which farmer can carry in his packet, or we can prepare a small capsule like in which the biofertilizer strains will be condensed, powdered formulation will be there. So the last two we are right now working in our institute, which the investigations are going on. So this is the timeline of biofertilizer development in India. So the earliest research was started in 1980, and in 1990 it was started as commercial inoculant. And IRI, Indian Agricultural Research Institute, started its first biofertilizer production as acetobacter and rhizobium, and followed by Tamil Nadu Agriculture University and so on. And then in 2000, the spread of the technology and formulations were gaining importance, and in 2010, so everyone talking about liquid biofertilizer, which is you can correlate it with the organic farming. So when 2010 the organic farming is gaining attention, now the biofertilizer is also gaining attention. So once everybody is aware of biofertilizer, then the quality is now concerned, and from that onwards, the biofertilizers transformation going very fast, and now, as I already told, it's NPK biofertilizers, seed coat formulations, and the powder or tablet-like formulations are in progress. Right, so this is the statistical data, which you can see. The green bar indicates the total biofertilizer production in India, and the small bar, which is adjacent, is our state, because I belong to Tamil Nadu, so I'm interested to collect the data. And if you take the annual production, it increases to a tune of around 12 to 15% annually in India, whereas if you take Tamil Nadu, its increase is nearly 23%. So as compared to other states, Tamil Nadu is little bit more progress in use as well as the development of fertilizer, I could say. Right. So if you see the total annual biofertilizer production state-wise, Tamil Nadu, Karnataka, Maharashtra shares nearly 50% of the total production, followed by other states. And in terms of liquid biofertilizer, Gujarat is progressing well. So this is the statistical data which you can get it from NCYF's website. Right. So I also look how much biofertilizer is required. So I calculated for my state, and you can do for yours. So what I did is I collected all crops that are grown for 1 year, and if I use all the crops as a liquid biofertilizer, how much kilotons or kiloliters of biofertilizer needed, and if I use carrier based, how much tons of biofertilizer that statistical work I did it for our institute purpose, and I did in 2005 as well as in 2018 data. You can see in 2005, if I calculate the total area, the biofertilizer accounts only 8% of the total area, whereas now when I calculated for 2019, it goes up to 26%. So it means that out of the total area, the 26% of the crops are being now biofertilized in Tamil Nadu. And if you calculate for India, it comes around 9 to 10%. So which has very high potential for the future. So this is overall scenario about the biofertilizer production in India, and now comes the science part. Right. So what is the principle of biofertilizer application? So biofertilizers are the preparations and that formulation, either liquid or carrier, that contains the viable cells, and when you apply, they should give some nutrients like nitrogen, phosphorus, zinc, potash, or some drought mitigation or against the pest and disease, something like that. But they should have some criterion to follow, right? So when you apply either through seed or seedling or soil application, it goes to it reaches around 10 power 3 to 10 power 4 cells per plant. It's all. So very, very minute quantity of cells of biofertilizer will be added to one plant, but that will not serve the purpose, and they have to colonize the entire rhizosphere or internal plant, whatever it may be, their preferred ecological niche, and then only you can say that you will have benefit from the biofertilizer. So for that, apart from its regular function like nitrogen fixation or P solubilization and so on, they should have these qualitative characters. So they should have a motility. Then only they will go and colonize the rhizosphere of the plant, and they should have very high carbon utilizing efficiency. So because the root exudates will produce more than 25 to 30 different types of carbons, including sugars and alcohols, and they have to acids. They have to have them as their carbon source, and they have to they should able to produce biofilm, so that they will exclude their competitors, and if they have antagonism, then they will eliminate them, and thereby they can give their benefit to the crop. So all I want to tell is, so the method of application or the strain what we are applying should ensure that that strain should sufficiently colonize the rhizosphere or any ecological niche of that particular plant in the young plant, then only they will give the nutrient uptake or drought mitigation or uh, pathogen resistance and so on. Right? So this is the principle behind the biofertilizer application. So whenever we formulate a product, then this the principle is very important for application, right? So now let me summarize what are the commercial biofertilizers right now available. So by functional wise, the first one is the nitrogen fixers. So we have symbiotic like rhizobium, associative symbiotic like azospirillum, and free living like aspergillus, and they have nitrogenous enzyme which fixes the which reduces the atmospheric nitrogen into ammonia, and by using GS Gogar pathway, that ammonia will be translocated and transformed into amino acids, and thereby they they will be added with nitrogen. So they can have the capability. So this table shows how much nitrogen this biofertilizers can give to a particular crop as a host, right? But when you apply this nitrogen fixing bacteria, there are so many important factors are there. The number one is the host plant, right? So it depends upon the which biofertilizer is applied to which host plant. So if you apply rhizobium to a cereal crop, then you won't get good nitrogen fixation, and vice versa. If you apply azospirillum, you won't get good nitrogen fixation in legumes. So they are highly host specific. And same way, if you use azospirillum lipophorum, it is mainly for rice, and azospirillum brasiliensis is for other other than rice like cereal crops. So uh, they have the tricks behind this, or the principle behind this host plant is its root exudates, right? And then the efficient strain. So there may be poor nitrogen fixers but efficient colonizers. Then if they were if high efficient colonizing bacteria with low nitrogen fixation, then they will be a burden to the host plant rather than a beneficial symbiosis to them. And also the soil biological attributes like total organic carbon, available nitrogen, and then the what moisture availability, redox potential, like many other attributes are also important. And of course, since it's a nitrogen fixers, the available nitrogen level is very critical. If it is very high, then they will not do the nitrogen fixation, and vice versa. And also the chemical synthetic fertilizers also against the nitrogen fixers colonization. So this is about nitrogen fixers, followed by mineral solubilizers. So the principle behind the mineral solubilizers there are two different principles are there. One is they produce enzymes to solubilize the organic form of that nutrients to release the insoluble forms like phosphatase. But that group of organisms, enzyme-mediated solubilization is very limited in biofertilizers, and this one is the organic acid production. So when they utilize glucose, that glucose will be periplasmically uh, ox- uh, uh, oxidized into gluconic acid and further oxidized into 2-ketogluconic acid by using the enzyme glucose dehydrogenase and gluconate dehydrogenase in the membrane. So if this happens, then the gluconic acid and 2-ketogluconic acids were excreted outside, and that acid will solubilize the insoluble form of nutrients like tricalcium phosphates, potassium silicate, zinc oxide, or like that. So by using this principle, many mineral solubilizers solubilize the nutrients and made it available to crop plants. So if they solubilize phosphorus, then you can call it as phosphobacteria, and if it is solubilizing potassium, then they were potash bacteria, and zinc as zinc solubilizer, right? So some organism, the zinc solubilizer may or may not solubilize potash, and the potash solubilizing bacteria may or may not solubilize the phosphorus or zinc. So that the principle behind the ore or the complex material specific solubilization is not yet fully understand. Please remember. So here you have a, a future thrust that how they choose their minerals. So some bacteria, some phosphobacteria will not solubilize zinc, and some potash bacteria will not solubilize phosphorus. Why? You don't have the answer. So those who are interested, you can search for the answers, right? And the main factors responsible for mineral solubilization is the rhizosphere. So they have to colonize the rhizosphere, profusely colonize, and then only they will do its function. As usual, the strain efficiency of the strain to gluconic acid production and excretion is very important. And of course, if it is phosphorus, the soil P; if it is potash, availability, and zinc, and so on. Again, the chemical fertilizers are not not favorable for these kind of biofertilizers, and they need a lot of organic carbon for their survival. So the soil organic carbon is a key issue. So decides the functionality of mineral solubilizers. And then the mineral oxidizers. So we have only one biofertilizer on mineral oxidizer, namely the sulfur bacteria. So it oxidizes the elemental sulfur into sulfate, and sulfate will be taken up by the plants. So in general, originally the sulfur coated urea like fertilizers were applied, and in soil also this bacteria are present, but they are all chemolithotrophs. But this mineral oxidizing biofertilizer is a organotroph. So uh, it's a facultative chemolithotroph, so they will use both organic and inorganic source for their carbon and energy. So thereby they can be mass multiplied under laboratory condition and you can apply. But the original chemolithotrophs are you can't grow them under laboratory condition. So here you can have it, and in our university we developed this as a biofertilizer and used for oil crops, because oil crops like sunflower, sesame, groundnut, they need more sulfur for improved oil accumulation in their kernel or in their seed. So for that you need huge amounts of sulfur, and now we have the technology to provide sulfur oxidizer as a biofertilizer. And then comes the hormones, right? So there are some biofertilizers. They will not be the nitrogen fixers. They are not the mineral solubilizers. They are not the oxidizers. But yet you can use them as biofertilizer. The best example is your PPF. So pink pigmented facultative methylotrope. It's a phyllosphere love organism. So they used to colonize the lower part of the leaf, and they utilize the methanol released through the water vapor during evapotranspiration, and that will be utilized and they colonize. And they are responsible for drought mitigation, and they have two different principles. They produce a lot of cytokinin, which is reduce the abscisic acid production, and thereby they close the stomata, and thereby you can reduce the loss of water. Whereas in the root system, they produce ACC deaminase enzyme, thereby they reduce the ethylene level. So by these two mechanisms, the PPFM is a very successful biofertilizer, and in TNU, in our institute, we produce this biofertilizer and extensively use it for drought mitigation. I hope you know Tamil Nadu is one of the states of India where you will find the temporary drought during agricultural system. So often for 1 month you won't get any rain. So if there is no rain, then the rice crop will not get because of Kaveri water. You won't get adequate water. So at that time we use this as a biofertilizer to spray on the surface of the leaf, and thereby we can protect around 15 to 20 days from the drought. And we did a massive demonstration in Tamil Nadu in 2014-15 to save the crop from rice crop from drought. Right, so this is another new technology which we developed from our laboratory, which is called stage-specific inoculants for rice. So in rice, we have 5 or 6 different biofertilizers we already documented, like Astrobacter, Phosphobacteria, Pseudomonas for plant growth promotion as well as biocontrol agent, and Phosphobacteria for phosphorus, potash for potash, and Methylobacteria for drought mitigation. And what we did is we apply all the 6 biofertilizers at their appropriate stage of rice crop, so that all the 6 biofertilizers will be like a package and use it, and we use it as a uh, very good inoculant for organic farming, where you won't use any chemical fertilizers and there won't be any interaction with synthetic chemicals. In this field, this stage specific inoculants come up very well, and we demonstrated all over in Tamil Nadu with the help of the Indian Council of Agricultural Research. They funded nearly 3 crore rupees for the entire Tamil Nadu demonstration, and we showed that it's a very good inoculant for organic farming. And another new uh, technology development in biofertilizer is NPK biofertilizer. So up to 2015-16, the biofertilizers were applied as one liquid inoculant for nitrogen, one for phosphorus, another for potash. Like individual strains will be present in one inoculant bottle. And after 2015-16, many research institutes developed this NPK consortium, and now the government of India approved the NPK biofertilizer as one of the consortium, and now the farmer want to use 3 different biofertilizer for 3 different nutrients, whether he will use only one inoculant which contains all the 3 in one, and he will apply. So this was first developed uh, by Indian Agricultural Research Institute in 2017, and now by using their patent product, many companies are nowadays producing this NPK biofertilizer, and now they have the quality standard for this NPK biofertilizer also. So they have shelf life of 1 year, and it has a separate protocol how to use all these things are now available to the farmers. And As I in the beginning of my talk, I told about the seed coat formulation and freeze dried formulation. So this we developed in our institute. So the main idea is the seed is the surface in which if you coat the microorganism by using a film, a polymer like film, thin film, and so that the microorganism will survive on the seed for at least 30 to 90 days. And once you sow it, when the seed get germinated, the root will get immediately about the biofertilizer without any problem. So the farmer won't use two different input during the sowing. So the seed itself will carry the biofertilizer. So for that, we concentrate since it needs huge amounts of inoculum to keep one seed of around 10 power 3 cells of biofertilizer cells. Then you have to concentrate the cells into very high load. So for that, we used industrial lyophilizer and spray dryers to concentrate the viable cells, and we also use different synthetic and semi-synthetic polymers to coat it, so that when that after the coating, the farmer won't feel in the hand any stickiness and so on. So by using this technology, the farmer, you don't need to advise the farmer to use biofertilizers because it is already coated with seeds and it will not have any harmful effects. And now we are using this technology to add again a chemical, so that chemical fungicides, like, so if they have chemical fungicide and if you add microbial cell, they have to separate by space by using a film. So in that sense, this technology would would be the third generation biofertilizer, I can say. Right. So now another strategy about biofertilizer application is biofertigation. So now, as I already told, Tamil Nadu is one of the states always a drought-prone area where most of the farmers now they change to drip irrigation for their agricultural fields. So during that time, they may feel difficult to apply biofertilizers or any chemical fertilizers. So we standardize the protocol to apply the biofertilizers through the fertigation tanks. So if you apply the biofertilizer in the fertigation tank, then they will travel through the laterals, and the drop wise they will evenly distribute to all the crop, all the plants, and thereby uh, you can be ensured that the biofertilizers can go and colonize in the roots of all crops. So we standardized for bendi, coffee, like commercial crops where the drip fertigation is extensively used in Tamil Nadu. And here we prepared a schedule for both chemical fertilizer as well as the biofertilizer. And the other uh, research, new trends that are available in biofertilizer technology is multifunctional biofertilizer. Why don't we go for one strain for nitrogen, another strain for phosphorus? Like now, many research institutes think that why don't we have a super strain which has to do nitrogen fixation, mineral solubilization, antagonism, drought mitigation, growth promotion, and so on. But to my knowledge, I could not find any such super strains are available. But the research is going on to maybe the candidates are Pseudomonas, like Pseudomonas fluorescens, Pseudomonas aeruginosa, Bacillus amylolecifaciens, Burkholderia vietnamensis, Streptomyces, like few candidates are available, but they were not yet commercialized as multifunctional biofertilizers. Another important ecologically niche organisms that are yet to explore is endophytes. They are all these biofertilizers so far used is a epiphytic organism except rhizobium. So they will go and colonize the rhizosphere region or rhizoplane region, and thereby they will benefit. But if you apply an endophyte, they will perform better than rhizosphere colonizing organism, because they will have less competition as compared to rhizosphere colonizing. So here again, Bacillus, Aeromonas, Micrococcus, Pseudomonas, like many candidates are available. But here the problem is there may be an differentiating the competitive endophytes to opportunist and passenger endophytes is little difficult. So ensuring that the endophyte should be a putative and very high colonizing throughout the plant system will be the bottleneck in endophyte based biofertilizer. And another is the quality assurance. Right now, the Bureau of Indian Standard earlier and right now, the Ministry of Agriculture, they developed the or FCO, Fertilizer Control Act, Fertilizer Control Order. They developed a few quality standards through which you can see how many viable cells are there, what is the pH level, how much moisture percent, and what is the uh, level of contamination. So only these four criteria will decide the quality of biofertilizer, which is not sufficient, because the mother culture which he or that company is using is very important. So for that we developed a technology called scar marker based quality standard. So we developed a PCR based protocol to identify whether the biofertilizer inoculum or the biofertilizer packet contains that particular strain or not. So that will tell that uh, directly tell how much uh, quality of biofertilizer is present. So this is the protocol. So what you have to do is you take the sample from the biofertilizer, take 1 ml or 1 gram, extract the DNA by using a 3 minutes protocol, and use it as a template for PCR, and you will get the results. So this can be used both for uh, qualitative check as well as like real-time PCR, how much cell load is present in 1 gram or 1 ml of biofertilizer. So this unique technology was developed by our institute, which is right now we plan to adapt for Tamil Nadu biofertilizer production units. So now the discussion is going on with our state biofertilizer production units. So this is a brief about the current trend and what next, what is the biofertilizer uh, production system is going on. So what are the significance of biofertilizer? So they assure that at least 5 to 15% of yield increase as compared to control, and they reduce at least 25% of their synthetic chemical fertilizer. And of course, if you use biocontrol agents, you can get rid of the harmful pesticides, insecticides, and antibiotics. And if you use fertilizers and biofertilizers, the fertilizer use efficiency get increased to a tune of 10 to 15%. And by using the continuous use of biofertilizers ensures the soil organic carbon, biomass, and enzyme activity of the soil, and it improves the overall soil ecosystem functioning, and of course the disease suppressiveness, and also resilience of the crop from biotic and abiotic stress. But if all these things are the positive side, if we use a good biofertilizer, and the constraints are too many, and because of poor quality it failed many times because the biofertilizer is not regulated properly. So the government of India or any government, they they encourage the biofertilizer production by reducing the quality standards. So due to that, a huge number of biofertilizer companies they started, and with poor quality and poor mother cultures. And so once the farmer used, immediately his feeling is not satisfactory because he has to observe that by using biofertilizer, he will get some benefits, whereas you won't get any visible differences. So he will get upset. So he will not use another time. And we have to update the farmers. Right now the knowledge about the biofertilizer to the farmers is very limited, right? And another most important is not timely available nutrients. See, if he want to sow the seed, immediately he can go to any agro service shop and he can buy. But if he want biofertilizer, he has to go for a little bit away where the biofertilizers are produced, and there only he can get. So it is the quality biofertilizers are not available in time. And as I said earlier, there is no strict regulations are available. And what strain that biofertilizer is being procured and used for mass multiplication, we don't have any adequate information, and we don't have any monitoring system, and there is no encouragement for biofertilizer usage by a farmer. So because of these constraints, still we have to go a long way to improve the biofertilizer benefits to the farmer as well as the crops. So for that, you have to follow three A's. So it should be appropriate biofertilizer. So you should not use inappropriate uh, mismatch host biofertilizer, and adequate quality. You have to ensure that the proper strain is there, proper cell load is there, there won't be any contaminants, and so on, and accurate method of application. So every time when the young plant, they should have adequate number of cells in their rhizosphere, then you will have good benefit. So this is about the significance and constraints of fertilizer. So I also how we can get rid of these problems. I just listed as farmers awareness. So the farmer, we have to educate the farmers about the biofertilizer, and what are the entrepreneurs' responsibility. So they have to make sure that uh, they are giving a good product to the farmers. So that's and they have to get trained for using biofertilizers, trains, mass production. At any time, there should not be any, at any stage of mass production, there should not be any quality deterioration we have to do. And same way for institutional responsibilities. So this is what they have to do. I just listed out, and maybe in another 3 4 slides I can conclude. So right now the biofertilizers what we are using is a single isolate study model. That means, so we go and collect some 20 or 30 strains of biofertilizer from rhizosphere or hydrosphere or somewhere, and screen them, and we will say that this is performing better, and then this will be evaluated under field condition, and then at 3 different locations or different states or different regions, and then we will say that this performs better and we can release it as biofertilizer. But we omit the real plant microbes interaction. So what next is that one? So we should understand the plant microbe interaction, then so that the most appropriate partner to that particular crop will be the next stage in the biofertilizer development. So we have to look little bit more appropriate, understanding the plant microbe interaction, how the plant root metabolites are there, and from that metabolites how the microbes utilize, and thereby how the soil get benefited. So if you understand the plant microbe and soils interaction, then from that if you develop an inoculant, then I'm sure that the biofertilizer technology will go in another uh, new dimension in the research. So many works were already developed. So this one is a very interesting paper you can see, which was published in Frontiers in Plant Science in 2017. So what they did is they developed after studying the plant microbe interaction, they identify one bacterium and they spray the bacterium on the flowers of the crop, right? So once the flower received that particular microbial inoculant, that that organism get colonized in the seed, and once the seed is sown to next generation, so during the germination of the seed, the inoculant which was already inoculated in the flowering stage get colonized, and it occupies the entire rhizosphere. So that kind of work we need for future biofertilizer production. So it refers the holobiont. Holobiont is the microbe, the plant and its associated microbiome. You treat them as a single ecological unit. Uh, and if you develop that kind of biofertilizer, then that will give more benefit as compared to the present single strain isolate methods, right? And it also, so this kind of approach we did it in our research institute. So we apply biofertilizer to improve the plant, right? So when you apply the biofertilizer, so it takes nutrients from the soil or from the atmosphere, gives to the plant, and the plant grows. And what about the opposite direction? Why don't every time when the plant grows, it excretes nearly 30% of its carbon in the form of root exudates? Why don't we use that carbon to improve the soil health? So that comes the plant soil feedback. So if we use a proper partner, so the carbon that were excreted from the plant can be transformed into a most significant soil carbon, and thereby you can increase the soil, and when the next crop comes, it will grow even without that particular biofertilizer. So that is the biofertilizer which you can come up with plant soil feedback. So feedback means it gives back to the first crops. The effect will be gives to the second crops, like that. So if it is positive and negative, so we can use this positive plant soil feedback through biofertilizer. That would be another new dimension research that is focus is needed for future. So that's all I want to deliver in this presentation. So to summarize my talk, the biofertilizers are no doubt that they are the best alternate agricultural input for sustainable agriculture, and they ensure macro and micronutrients, particularly nitrogen, phosphorus, potassium, zinc, sulfur. These five nutrients we have, and of course IAA like growth hormones also. And some biofertilizers will ensure biotic and abiotic stresses, and they also induce systemic resistance and tolerance. So nearly 10% of the agricultural crops of India are nowadays using inoculants, which has to increase extensively. And by using this technology, you can reduce nearly 25% of the synthetic fertilizer. Thereby, you can reduce uh, the cost of production cost. And nowadays, because of the organic agriculture and the human health, this biofertilizer is gaining attention. So the main issues are the ineffective strains, lack of proper authentication of the strain, and the soil and environment mediated failures. The moisture stress is another one. If the soil doesn't have enough moisture, then the biofertilizer may not function. And lack of high quality standards and awareness are the major constraints which the R&D units and entrepreneurs should focus. And the upcoming biofertilizer technologies are endophytes, stage-specific inoculants, car markers, and of course rhizosphere engineering. I didn't touch rhizosphere engineering for want of time, which is another field through which you can improve the biofertilizer use efficiency by using above ground strategies. You can spray some of the hormones so that they will excrete more amounts of carbon, and thereby the biofertilizer can increase. So that is also possible in future. And of course, to all these things, we should understand a good plant microbe soil interaction. And I once again thank the organizers for giving this opportunity to share my experiences and views about biofertilizer. Thank you.
Thank you, sir. The session is now open for discussion.
Yes, Dr. Nandakumar.
Yeah, congratulations. You have done a lot of work. I think more than a century, the university is working in Tamil Nadu
Yeah.
and South India.
Yes, sir.
We
Thank you.
used to meet lots of scientists from Coimbatore for various conferences of Mycological Society of India,
Yeah, yeah.
and we learned a lot of new techniques from them. I was hearing your presentation on comprehensive presentation. Congratulations
Yeah.
for giving in nutshell, I think almost everything that we are supposed to know on biofertilizers. Sir, accept our compliments on behalf of State of Goa for contributing to green revolution in Tamil Nadu in particular and South India in general.
Okay.
We congratulate you for this work. My
Thank you.
specific question is very simple, sir, and it's related to nutrient deficiency, like say iron and molybdenum, which is related to activity of nitrogenase and nitrate reductase.
Yes, sir.
What we observed during our general observation, it's not published, that when we were working on Mimosa pudica as a sort of a model leguminous plant
Yes.
because of the nodules,
Hmm.
it was our observation over last 10 years that the nodulation is very deficient in iron and molybdenum deficient soils. Understood? Hello.
Yes, sir. Yes, sir.
And in other areas, we found also where molybdenum concentration exceeds a certain, that concentration, the nodulation is also not good. It looks like higher molybdenum concentration is also detrimental to nodulation. So, whether you give seed treatment with molybdenum along with rhizobium bioinoculants, that's what I wanted to know to
Yes,
improve the
sir.
efficacy.
Yeah, I can answer this question. Can I answer
Yeah,
now?
sir, please go ahead. Yes.
Yeah. Yeah, thank you for your nice words. So this kind of issue is going on in our area also. In one of patch of Tamil Nadu soil where we have red laterite in which iron toxicity is there, so that the molybdenum is very low. So
Correct.
what we did is uh, we during the sowing we can't use molybdenum to treat the seed. So
Correct.
what we did is we attempted two methods. One is we prepared a micronutrient mixture which contains very trace amounts of molybdenum, boron, and a little bit of zinc, and we made it like a mixture and we apply through soil, right? And the
Yeah.
same three combinations we also sprayed on the surface of the plant along with the DAP at 25 days after planting. So both performed well. So that technology you can try. So you can apply as a soil these three micronutrient mixture during sowing, or you can spray them on the fluid on the surface of the plants.
So,
Both
sir,
ways you can get.
yes, sir. Some, sir, other places they have tried with chickpea and cowpea, the application to the seeds
Mm,
along
mm.
with biofertilizer. What do you have to say about that? Chickpea and cowpea, they have applied the seeds, molybdenum solution.
Yeah, but in our it has the practical difficulty in our state, so that's why we didn't try about seed treatment of micronutrients. But if it works, then you can go for it, no problem, because the concentration of micronutrient, molybdenum and boron, they are very little. They will not affect our
Correct,
biofertilizer strain. So
correct, correct,
you,
correct.
you
Thank
can
you, sir.
go without any issue.
Thanks,
Yeah,
thanks.
yeah. Thank you.
Any more questions?
Yes, Dr. Sheila Paul. Please unmute and ask your question. Miss Sheila? Yeah. No, it's in mute. Please unmute. Miss Sheila, please unmute.
Ma'am, you can type your message in the chat box if you're unable to
Now
mute
she
your.
now she unmuted. Please. No. Dr. Trupti, she had some issue with the unmute problem.
Yes, sir. Ma'am, you can type your message question in the chat box. I will read it for you.
Dr. Trupti, she had some problem.
Yes,
I
sir.
could
I
not
think
see
she
any
has some
message
problem.
also.
Yes, sir. Any more questions?
Also 30 रुपये IBS.
So, how about using microbiome for biofertilizer to improve the microbiome around the plant so as to enable better nutrient uptake?
Yeah, it is a very big question, which you have to work a lot. Right now, we didn't attend that kind of approach, but that's what my last two slides indicates, right? So when you introduce through seed or soil, when the crop grows, the introduced inoculant should cover the entire microbiome, entire rhizosphere. Right? Theoretically, you can talk, but in practical, it is not possible right now. So that's the holobionts based biofertilizer approach. So we still working on it, but it's a very big question.
Because I even read about this improvement or engineering the microbiome
Hmm.
to improve the carbon mitigation.
Yeah, you can
So a
do.
lot of that work is also being done on this for engineering the microbiome
Yeah, actually,
around the plant.
originally, why what they did is mainly they modify the metabolic pathway of that particular microbe, and when you
Mhm.
introduce to the crop, so they will scavenge mostly they use it for bioremediation. So this
Yes.
engineering aspect is mainly extensively used for bioremediation, not for
Okay.
carbon sequestration. Carbon
Okay.
sequestration, there this kind of approach is very limited
Okay.
because particularly like copper, cadmium, in
Mm-hmm.
that sense, there are extensive works are going on. So
Okay.
they inoculate, and when you have a very good plant microbe interaction, so the
Mm-hmm.
engineered rhizosphere will grab more amounts of heavy metals, right?
Okay.
And thereby, it will deposit in the plant, and you can pull out the plant and you can incinerate, and thereby you can avoid the. It's an in situ remediation. So that
Okay.
approach worked well through this rhizosphere engineering, but I didn't see any soil improvement like carbon sequestration. That we have to go a long way.
Okay. No, I was attending one conference and there somebody was talking about this particular aspect of it, and that
Okay,
is where I,
okay.
uh, whether
Yeah, it
something
is a
similar is being
very
done over here.
yeah, it's a very good approach, but
Yeah.
we didn't have basic data for that.
Okay.
For which
Okay.
you have to go a long way. You have to
Okay.
need sophisticated equipments and facilities for the understanding the rhizosphere functioning and so on. And we are working on that field also, but it's
Okay.
very early to give any conclusive results to you.
Okay.
That's why I
Okay. Thank you. Thank you, sir.
thank you.
Any more questions?
Yeah, I have one more question for
Yes,
Dr.
sir.
Balchandar.
Yes, sir. Please.
Since we have some time, I am using it because you see, this is a river of knowledge which comes to our university. We are not agricultural state.
Yes, sir.
We
No
are highly
problem.
urbanized, and whatever
Yeah.
parcels of agriculture are left, they are converted into real estate. So we are looking to Tamil Nadu and other states for now all these leads. You will not see any agriculture in Goa after 5 years. So we may do hydroponics and things like that and terrace gardens, but you will not see the conventional agriculture. My question is very simple, sir. I have done some work on earthworms from different angle.
Okay.
My question is, how, sir, your experience of application of biofertilizer shows about increase, decrease, or whatever in natural earthworm activity in the soil in which you apply the biofertilizer? Whether it is conducive or it is whatever it is, means it is
Sir,
same, or you see that it is maintained and utilized effectively, because earthworms also play a very important role in
yes, sir.
soil, the environment, I believe. Do you have
Yeah.
any type of work on that in that area, sir? Earthworm
No,
ecology and
sir.
biology.
No. Sorry, I could not give any good answer for this because biofertilizer versus earthworm, we don't have any practical experience. But theoretically speaking, uh, there is no connection between the earthworm population and biofertilizer because biofertilizer functioning is entirely different with our earthworms ecosystem. Maybe we can say that if we use more of biofertilizer, we can reduce the chemical inputs and thereby we can increase the earthworms livelihood in the soil. So that may be the one line answer I can give.
Thank you, sir. On behalf of Department of Microbiology, Goa University, I express my humble thanks to Professor D. Balachandran for the informative talk on recent trends in biofertilizer technology. We are all aware of the fact that use of bioinoculants is the need of the hour for sustainable agricultural practices. As said by Sir, there has been some awareness among the farmers, and the area which was utilized as 8% in 2005 has increased up to 26% in 2018. So, I'm confident the participants are benefited by the knowledge shared by you on various aspects of biofertilizer technology, including the principle, working, various types and their compatibility with host, different types of formulations, use of biofertigation and multifunctional biofertilizers, and also the constraints faced in this area. We are grateful to you for sparing your invaluable time and enhancing our knowledge in biofertilizers. Thank you, sir.
Yeah. So, can I say a few words? Yeah, thank you for your for giving this opportunity, and I wish to have a good 3 days seminar, and hopefully the students and research scholars will get benefited through various talks. I have seen the list of presenters and the topics which were very good spectrum you covered, and my all the best to the organizers to as well as the participants to get good opportunity to improve the uh, knowledge in microbiology, and my best wishes and thanks once again to Dr. Sandeep and their team for uh, giving this opportunity to me. Thanks. Thank you to one and all. Thank you.
Thank you, sir.
So, shall I stop sharing?
Yes, sir.
Okay. Thank you. Goodbye.
Goodbye, sir.
I thank all the participants for attending the sessions today. We will close for the day and meet again tomorrow at 10 o'clock. Thank you.